Good evening, students. Today I am going to discuss about the next topic of this chapter. The topic name is multiple allelism. See, what is multiple allelism? Commonly in organism's body, two alleles control the all characters. But when we observe the human blood groups, majorly three alleles controls the human blood groups. So multiple allelism means three alleles controls the characters. See, what is multiple allelism means three alleles controls the characters. For example, human blood group. Human blood groups. In individually, two alleles controlled by the blood group, and also when we are going to population, three alleles controls the human blood groups. See how they control. What are the alleles means? See, I will draw here chromosomes. So these are the chromosomes. Already you know very well, inside of the chromosomes consist of double stranded DNA. See, this is called double stranded DNA. So this is called chromosome. Inside the chromosome consists of DNA. Then in this what kind of alleles or what kind of factors are controlled by the human blood group means majorly three types. What are they? One is A allele, and second one is B allele, and third one is I allele. See, three types of alleles control the human blood groups. So this kind of method we are called as a multiple allelism. Okay, then we are going to one more important content, that is called pleiotropy. Pleiotropy. See, what is pleiotropy? Pleiotropy means two alleles controls the more than characters, more than one characters, not a single character. Such kind of character is called, such kind of process is called pleiotropy. So what is pleiotropy? Two alleles controls the more than one character, more than one character. So such kind of process we are called as a pleiotropy. What is pleiotropy? Two alleles controls the more than one character, not only one character, like height, not only height, or shape of the seed, not only shape, like that. In pleiotropy, it controls the two alleles controls the more than more than one character. Okay, I will give two examples for this pleiotropy. Okay, first I am going to give one example in a hemoglobin, where the hemoglobin is present. Hemoglobin. So the hemoglobin present in the blood. So when we observe the hemoglobin, it consists of nearly 4 heme molecules and also 4 globular protein chains. I will draw roughly for understanding purpose. See here, for suppose these are the 4 heme molecules. What are they? Heme molecules. So how many? Number 4 number of heme molecules, and also it consists of 4 globin protein chains also. So it consists of chains. Each heme consists of one globin chain. This is one globin chain, and this is one globin chain, and this one also one, and this one also one. So these globin chains, how many globin chains? Globin protein chains. Globin protein chains. Nearly 4. 4 number of globin protein chains. Okay. The first, these two chains are called alpha chains. So what is this? These are the alpha chains. These are indicates alpha chains, and these two are called beta chains. Remember, 2 alpha chains and 2 beta chains. So these globular proteins consist of 4 protein chains. What are they? 2 alpha and 2 beta. 2 alpha and 2 beta. Okay, coming to the beta chain. Here some changes we are noticed, some mutations we are noticed in the beta chains. See what kind of mutations means? Already it is a globin protein. What is this? This chain indicates what? Globin protein. Globin protein. See, already you know very well, the proteins are made up of some subunits. What are the subunits means? They are made up of amino acids. See, these are the chain of amino acids. See, like this. So this is called amino acids or amino acids. So in this amino acids, mainly the 6th amino acid, commonly it releases glutamic acid. So this is the 6th amino acid. 6th amino acid, commonly there is no mutation in the genes. The amino acid, 6th amino acid, it releases the glutamic acid. Glutamic acid. Due to the some environmental changes or some mutations which are occurs in the DNA, the glutamic acid is not released. So one more, the different substance is released from the 6th amino acid. So what kind of substance it releases? When it get defective, then it will be defect by the environmental issues. So when the amino acid defects, then it release the valine. It release the valine. Understood? Commonly, the sixth amino acid in the normal DNA, the sixth amino acid release the glutamic acid. During the mutations, if any mutation will happen, if the gene will be damaged due to the environmental conditions or UV rays, then the glutamic acid is not released. Instead of glutamic acid, one more substance is released, that is called valine. So when the valine is released, here some changes we are noticed here. What kind of changes means? The hemoglobin, which type of hemoglobin will form means a defective hemoglobin is formed. A defective hemoglobin is formed due to the release of valine instead of glutamic acid. Then this is one type of change. One character is changed here. Then what is the second character means? The shape of the RBC is changed. That means normally RBC shape, which kind of shape it have? It has majorly biconcave shape, like this. The RBC cell is look like a biconcave shape. Then here the RBC shape is changed and it converted into, or it modified into the sickle shape. Which shape? Sickle shape. So these kind of sickle shape RBC which are present in the human, and it leads to one genetic disorder in the humans. What is the name of that genetic disorder means? Sickle cell anemia. Sickle cell anemia. See, very important. Sickle cell anemia is a one genetic disorder. Genetic disorder. Remember. So mostly this disease we will found in African countries. In African countries. So this is the kind of two characters are controlled here. See, what are the characters controlled by the factors? One character is hemoglobin, damaged hemoglobin, or the hemoglobin is converted into defective hemoglobin. What is the second character controlled by the gene? The RBC cell shape is changed and it converted into sickle shape. This sickle shape leads to one genetic disorder. The genetic disorder name is called sickle cell anemia. So mostly this disease seen in the African country people. So like this, here two characters are controlled by the gene. What are the two characters? See, one is hemoglobin, defective, normal hemoglobin is converted into defective hemoglobin. Then normal RBC shape changes into the sickle shape. So this is the one example we are seen in the humans. Okay, now coming to the second example in pea plant. Okay, see example 2. Example 2. In a pea plant, I will take a pea plant. So, in this pea plant, here also you have to remember the pleiotropy seen in human hemoglobin and also pea plant also. So, what is the example? What kind of changes we are noticed in the pea plant? See, first I will take the chromosomes which are present in the pea plant. See, here I will take 3 pairs of chromosomes. See, here I will take 3 pairs of chromosomes. Already you know, inside of the chromosome consists of double stranded DNA. See, this is called double stranded DNA. Okay, why I will take here 3 pairs of chromosomes? Normally 2 pairs of chromosomes. We will see in all characters they are expressed by the 2 pairs of chromosomes. But why I will take here 3 pairs means it is a different process. What is this? Pleiotropy. Pleiotropy means what? So, it controls the more than characters, more than one characters. For that reason, I will take here 3 pairs of chromosomes. See here, for example, I will take the seed shape and size of the starch grains. So how the chromosomes or DNA is controls the seed shape as well as inside of the grain, starch grains. See, for example, I will write here 3 types of allele. See, first chromosome having capital B allele and second one is also capital B. So, these are the pair of alleles which are present in the DNA like this. In the DNA, 2 types of alleles are present. What are they? Both are capital B. Then, in the second pair of chromosomes, it consists of one is capital B allele and second one is small b allele. This is different type. And third type of chromosomes, it consists of the both small b alleles. This is also small b, it consists of small b, small b. See, these kind of alleles belongs to the pleiotropism. It controls the more than one character in the pea plants. How they control the more than one character? See, now I will draw here. See, first I will take the capital B, capital B. These are the 2 alleles. So from this, which is dominant? Both are capital. So capital B indicates it is a dominant character. So I will take example for the seed shape. Seed shape. This allele control the seed shape. So which kind of seed it forms means a round seed. See, which type of seed it forms? A round seed. So it is a round seed. It is a round seed. Why it Round seed means B is a dominant trait or dominant character. So dominant always round, not wrinkle. Remember, round is always a dominant character. In the first pair of chromosomes, both alleles are capital B. Capital means dominant. So the dominant genes or dominant allele, it produces the round shaped seed. Remember that. Why round shaped? Always the round shape is a dominant character. Okay, coming to the second one. So when we observe the second pair, capital B and small b. So capital B, small b indicates which is dominant here. Here B is also present. So here also it is also dominant. Capital B is also dominant. So it also forms the round seed. So this is also which type of seed it is? Round seed. See, it is also round seed. Remember. Okay, coming to the third pair of chromosomes. Here, which type of alleles are present? Both are recessive alleles. So the recessive alleles, they are not able to produce the proper shape. For that reason, this pair of alleles, they forms the wrinkle seed. See, like this. They forms which type of seed here? A wrinkle seed. Wrinkle seed. So this and all shape of the seed. This shape of the seed is controlled by the these alleles. So here how many alleles? Capital B1, small b1. So this type of alleles controls the one character of the pea plant. What is that? It controls the shape of the pea seed. Okay, and it also controls the one more character in the pea seed. What is the one more character means? That is called starch grains. Size of starch grains. Size of starch grains. See, inside of the seed consists of a granule-like arrangement. Those granule-like arrangements we are called as a grains, like this. See, in the first seed, both are capitals. Both are capitals means here large size granules are present, starch granules. So, which type of starch granules it produce means large size starch granules. Large size starch granules. It produces the large. Why large size? Here both alleles are dominant. Dominant, it is responsible to formation of the large size. So large size is always dominant character. Okay, coming to this. Here capital B, small b, two different alleles are present. But what kind of starch grains are produced inside of the seed means here B dominant. So here also large starch grains are formed. No, that is wrong. Here one important process we will follow here. What is that means? See here B allele, capital B and small b. So capital B here produce the some amount of protein also. Here recessive, there is no protein. There is it have a defective allele. See, this is defective allele. this is dominant, but this dominant allele, it is not produce the 100% of protein here. For that reason, here one specialization we are noticed here. What is the specialization means? Here intermediate starch grains will form here. Which type of starch grains? Not a big, not a small. So intermediate size starch grains are formed by the second pair of alleles. Okay, coming to the third one, both are recessive. Recessive means they forms small size granules like this. They forms small size granules. Small size granules. So this kind of process we are seeing in the pea plant. This is called Pleiotropy. See here, capital B, small b are two alleles. They control the more than one character in the pea plant. What are the two more than one character? One character is seed shape. It controls the seed shape. It is one character. And the second character is size of the starch grains. So this is about the Pleiotropy which is seen in the pea plant. Okay, take a screenshot. Now I will start the Mendel's second law. See, Mendel's second law is very simple. Okay, recall one time Mendel's proposed three laws. What are they? Law of dominance, law of segregation, and law of independent assortment. Already first law completed, law of dominance. Now coming to the second law, Mendel's second law. Mendel's second law. See, what tells about the Mendel's second law means the second law is a law of segregation. That is called what? Law of segregation. See, what is the meaning of segregation? Segregation means divide. So the alleles are divided into to form a new gametes or a gametes. That is called segregation. Segregation means what? Dividing or divide. Segregation means what? Divide. So I will give one example for this law of segregation. For example, if you know, if you take one pollen grain, how the pollen grain will form? See, pollen grain means already you know. So this is the stamen of the flower. See, this is the stamen of the flower. The stamen consists of anther and filament, two parts. And the anther consists of male cells here. These male cells are called pollen grains. Pollen grains. See how the male cells or pollen grains are formed in the anther. So where it starts means they are formed from the pollen mother cell. Remember. So the pollen grains are arises from pollen mother cell. Pollen mother cell. See now I will draw the rough diagram for understanding purpose. For suppose this is the pollen mother cell. What is this? Pollen mother cell. So inside of the pollen mother cell, it consists of nucleus. Already you know inside of the each cell consists of nucleus. So this is the nucleus. So inside of the nucleus consists of chromosomes. See, for suppose these are the chromosomes. See, these are the chromosomes. So inside of the chromosomes consist of DNA. See, this is called DNA. DNA consists of genes, and also the genes consist of alleles. Okay, here for suppose one gene, one part of the DNA here, one part of the DNA in the chromosome, it consists of T allele, capital T allele. The allele name is what? Capital T. And the second chromosome, this allele is a small t. Remember, these are the two alleles which are present in the DNA. And this arrangement we are seen in the pollen mother cell. Then these two different alleles, they start to producing the two gametes here. What kind of gamete? From this chromosome, capital T, this is one gamete, and this chromosome, small t, this is one gamete. So here two gametes are formed. Two gametes are formed from the alleles. From the alleles. Remember, here this is called the law of segregation. So the chromosomes which are present in the pollen grain, pollen mother cell, the alleles, it segregates the two gametes. Alleles it segregate into to form the two gametes. So like this, here chromosomes, what kind of activity chromosomes are controlled here means the chromosomes, they segregate their alleles into the next generation. That means these alleles divide into, it forms the two gametes. So the alleles are segregate to form the gametes. Such kind of process is called law of segregation. Understood? So this is the example of the Mendel's second law. Okay students, are you clear? Okay, if you like my video, please subscribe my channel and share my video to your friends and relatives. Okay, thank you very much.